Perfect. I think we are on. All right, perfect. Hello, everyone, and welcome back to Ask a Scientist with the Asheville Museum of Science. My name is Abby, and today we will be talking to Dr. Jocelyn Colella. She is a postdoctoral researcher with the University of New Hampshire. She is also the incoming curator of mammals. Yeah, is that right? At the University of Kansas Biodiversity Institute. She's been published in a number of distinguished journals such as Nature Communications Biology. And her research focuses a lot on genetic evolution and how our understanding of genetics can help support conservation efforts. Don't forget to go ahead and send in your questions. We will get them answered as we move along. And thank you so much for joining us today, Dr. Jossie. I will pass it off to you.
Cool. Thank you. Let me share my screen. Cool. Hi, everyone. Thanks for being here. I wanted to start with a little story. You know, the other day, I was out fishing with my dad, just last weekend, actually, and we were talking about the impacts that our fishing has on fish in the oceans and the rivers. I should probably be honest, though, our fishing didn't actually impact the fishing at all that day because we didn't catch anything. But assuming that other people were more successful and caught more fish, we were wondering, you know, they base how many fish we can catch based on how many fish are out there. And my dad said, how can they possibly know how many fish are out there? And I said, Dad, they don't just count fish. They're not like 1, 2, 3, oh, there goes a whole school of fish. Oh, there was a blue one. Does that count? That one's really deep. That wouldn't be very accurate. We instead use genetics to count fish. We can use genetics to estimate how big a population is. As I was introduced, I'm Jocelyn Colella. I'm a postdoc at the University of New Hampshire, and I'm a mammal geneticist. And I will be starting a job at the University of Kansas this next year. Today, I'm going to share with you how we can use genetics to improve public health and save animals, including fish. We're going to talk about 4 different things today. First, genetics. What is it and how does it work? Then, once you're all experts, I'm going to show you how we can use genetics from one fish to see how many fish are in the river closest to your house. Then, we'll see how scientists use genetics to figure out where different diseases and germs come from. And finally, we'll finish by learning a little bit about genetic editing, where we can intentionally change a piece of DNA to change something about an animal. So let's get started. What is genetics? Genetics is the study of DNA. And DNA is this twisty red molecule that I've shown here. And DNA is just like a recipe to make an animal, the exact same way that we have a recipe to make cookies. If we combine the right proportion of sugar, flour, eggs, chocolate chips, we get chocolate chip cookies. And DNA contains all the instructions and the different ingredients to make an animal. If we change our cookie recipe a little bit, say by adding these little blue instructions here, then we may go from having chocolate chip cookies to having fortune cookies. And the same thing happens with DNA. A couple changes to the recipe, and we may now have a cat instead of having a dog. So how do these instructions work? Say our recipe tells us to add chocolate chip cookies and bake at 400 degrees Fahrenheit. If we look at DNA, this red molecule, the little blue guy that I've shown here is a nucleotide, which is basically like having one letter in our cookie recipe. If we have 3 nucleotides in a row, all 3 of these blue guys, that's called a codon. And a codon is basically like having one word in our recipe, chips. It's a common misconception that the codons across from each other talk to each other, but really they don't. They're only stored this way to save space. DNA contains a lot of information. It's a big recipe, so we need to shrink it down really small. and this lets us save space. When reading the recipe of DNA, you only read one half at a time. For example, the blue half of this recipe may be talking about how to bake chocolate chip cookies, which is what we want. But the yellow half may be talking about making pumpkin pie. And those are very different, so we don't want to get them mixed up. So we need to only look at half the recipe, just the chocolate chip cookie one. Remember, DNA is twisted, so that's kind of why this half looks a little bit weird. When DNA is split in half, the recipe is called RNA, and that's just a single recipe, just chocolate chip cookies. In RNA, two codons next to each other are read like two words in a recipe. So here we have chocolate chips, chocolate in yellow and chips in blue. As we continue to string words or codons together, we eventually get our full recipe. This says add chocolate chips, bake at 400 degrees Fahrenheit. Easy as that. This is just one example of one sentence in one recipe. But the recipe to build a human is 3 billion nucleotides or letters long. If we divide 3 billion by 3, because there are 3 nucleotides or 3 letters in one codon or one word, that means the recipe to build a human is 1 billion words long. That's a lot of words. That's 300 times the number of words that are in all the Harry Potter books combined. It's massive. So where is all this information stored? It's actually stored inside of a cell, a tiny, tiny cell that's less than 1 millimeter in size. The cells are what make up all of our different tissues: lung tissue, heart tissue, liver tissue, all of it. And if we zoom in using a microscope, we can look closely at a human cell. So here on the left, we've got our human, and then right next to him is this cell. And the DNA is packed into the nucleus of the cell, the small middle part of the cell. And it's kind of wound around these little X-shaped structures called chromosomes. We can unwind the DNA from chromosomes, kind of like unwinding a spool of yarn. And once it's unwound, scientists can look at special sections of the DNA that we call genes. We're interested in genes because these are the pieces of DNA that make proteins. Proteins are the building blocks of tissues. They're literally called the Legos of life because they can combine in a bunch of different ways to build different structures or different tissues, like lung tissue or heart tissue or skin tissue, all of that. But let's take a step back. We're going to look a little closer at genes on chromosomes. All mammals, including us, have 2 copies of each chromosome. So remember, chromosomes are these big X things. Inside of them is all of this DNA wrapped up like yarn. And we have 2 copies of each. And that means we also have 2 copies of each gene. 1 copy from mom and 1 copy from dad. Hi, Mom. That way, if 1 copy doesn't work for some reason, we have a backup that can make up for it. Those 2 copies can either be the same or they can be different. For example, if we look at the gene for eye color, if both of your parents have blue eyes, you and all of their children or all of your siblings will inherit 2 copies of the blue eye gene, 1 copy from each parent. Therefore, you and all of your siblings will have blue eyes. But if your dad has brown eyes and your mom has green eyes, you will inherit 2 genes that code for 2 different eye colors, brown and green. And that means you could have brown eyes, you could have green eyes, or you could even have hazel eyes. When the 2 copies of a gene that you have are different, scientists call this being heterozygous. This really big word up here that just means different. And if the two copies of a gene you have are the same, like we saw in the blue eye example, we call this being homozygous. Let me make sure you can see that. Organisms that are closely related to each other typically have the same or very similar DNA, making them more homozygous. Although it's a little more complicated than this, I think you get the general idea. Getting back to our fish, let's look at this example. If we have a small population of fish, there's only a few here, it's possible that the fish in our population may be all red, just by chance. Different shades of red, sure, but they're all red fish. The fact is that, the fact that they're all red means that they're probably all closely related. And because they're all red, the only color gene that they can pass to the next generation is also red. Because a red mom plus a red dad equals more red. So the next generation of fish will be similar to this one and likely also be red, unless of course a new color or a new mutation pops up. In contrast, if we look at a larger population, here we have one fish, two fish, red fish, blue fish, and also some yellow fish. In this case, when a red, yellow, and blue fish reproduce, they can make a bunch of different colors. Not just red mating with red fish to make more red fish, or blue with blue to make more blue, but also yellow can mate with blue to make green, a color that didn't even exist before. And blue could mate with red to make purple, another new color. And this can make all sorts of different color combinations. In this large population, most of the next generation of fish, all the new fish, will have two different genes that code for two different colors. That means they will be more heterozygous or more different. We can use this information to predict the size of animal populations. If we look at the DNA of only one individual, this little yellow guy, if both the color genes from his parents are different, we know this fish came from a large population. If both the color genes are the same, he probably came from a small population. And this is exactly what I do. Sure, it's a little less exciting when you look at it this way, but it shows us that swordfish populations were made up of 15,000 fish, or 15,000 individuals, about 10,000 years ago. And since then, the population has decreased or gotten smaller more recently. And as scientists, we can use this type of information to figure out how many deer we should or shouldn't hunt, and how many trees we can cut down to make paper and build houses with, or how many fish it's okay to catch. So I told my dad, we don't just count fish. We use their genes to figure out how many fish are out there. And that's why if you want to go fishing, you need a fishing license to make sure that we don't catch too many fish. There are only so many fishing licenses given out at one time, so we can make sure that we have healthy animal populations going into the future. And I think we have a question.
Yeah, so would you say that tracking a population with DNA is more or less accurate than doing something like marking and recapturing?
They are both great, and scientists use both. They tell us different information, so you can tell if a population is growing more quickly if you're doing trapping or mark and recapture type sampling. But you can tell more about the history of a population looking at their genetics. It's a great question. So in order to do all of this, I need samples, right? In order to do genetics, we have to have samples. And it's really hard for scientists to be sampling everything, everywhere, all the time. After all, I'm only one person, and there's only so many people who do what I do. So we need people like you to help us build these libraries of life. If you hunt or fish, or maybe you know people who do, Or perhaps you've even just found something really cool like bones while you were on a walk in the woods. You can reach out to your local natural history collection to see if they want samples from your area. Interestingly, common species like mice or raccoons are often underrepresented in museum collections. So while you might think it's boring that you caught a mouse in your house, it may be an awesome addition to a museum collection. Be sure to reach out to the museum before sampling so that you know what kinds of details the museum's going to need, like where you found the animal and when. And we can then use these data to estimate population sizes. And this is really important because large populations really are better. Large populations automatically have more diversity, which is good because diversity improves survivability or survival. We've got another question.
Yeah, so Travis asks, do you need an initial population reference count or, to do DNA population analysis?
You actually don't. You do not need an initial count, which is really cool. It's because of the way DNA is inherited, you can get an estimate without ever knowing how big the population was originally, which is why diversity is awesome. So diversity is a really, really good thing. And it allows us to predict what animals will be better at surviving under potentially different situations. Like I said here, if we had a disease that came up that only targeted redfish, the small population here would go extinct. They would all get wiped out, which is a total bummer. While in the large population over here on the right, the redfish still might die, but because the population has more genetic diversity, the blue and the yellow fish would be able to survive and reproduce, and the entire species would survive. The same thing is true for humans. Just like we saw with coronavirus is a new disease for humans. It's also the reason we're all stuck at home, the reason I'm presenting to you on Facebook Live, and why we have to wear masks around town, wash our hands, and stay 6 feet apart. And honestly, it kind of sucks, right? So naturally, we want to know, where did coronavirus come from? And how did it get into humans? And can we stop it from happening again? Again, we can use genetics to answer these questions and to track down where coronavirus came from. Because viruses have DNA too, kind of. They have a smaller version of DNA called RNA, which we saw earlier is basically half of a piece of DNA. The small size of RNA allows coronaviruses to copy themselves really, really fast. To figure out where our coronavirus came from, we can compare the RNA of human coronaviruses to the RNA of coronaviruses found in other mammals. Yep, you heard me. Humans are not the only ones that get coronaviruses. There are hundreds of other animals that get coronavirus. Sometimes we just can't even tell that they have it. The RNA of bat viruses or mouse viruses or even pangolins, this cool scaly guy here, or pigs, can tell us about their virus, and we can compare it to ours to see how different they are. Because DNA and RNA mutate or change a little bit over time through the process of evolution, each strand or each virus will be a little bit different. Both DNA and RNA have to make copies of themselves to make new tissues. It's kind of why you have to cut your fingernails or get a haircut every once in a while, because you're making new tissues. And every time DNA is copied, there's a chance for little mistakes to be made. And those little mistakes are called mutations. Now, I wish that mutations gave us superpowers, but usually mutations cause no obvious change. Womp womp. They just kind of exist as genetic diversity that's in a population and can be passed to the next generation. For scale, each human is born with about 60 new mutations. So you have 60 mutations that neither of your parents have, but you still probably look a lot like your parents. That means that because the human genome is 3 billion nucleotides or 3 billion letters long, that means there's a mistake about every 50 million letters. And that's really not very many. Think about it. If I had to handwrite the entire Harry Potter series 300 times, I would probably make a lot more mistakes. So mutations don't occur very often. The more similar that two DNAs or two viruses are, the more likely they are to be closely related. To compare RNA viruses between humans and other animals, we have to go out into the wild to get samples. And this is the super fun part of my job, where we get to travel all over the world and work with all sorts of animals of all shapes and sizes. This is me holding a bat in Costa Rica, and this is one of my field crews collecting data in Panama. Once collected, these samples are stored in natural history museums. Yep, these museums do way more than just give tours. They're super important for research and genetic research. This is a behind-the-scenes view of the frozen tissue collection at the museum I work at, the University of Kansas Biodiversity Institute. Inside each of these giant metal vats are columns full of little white boxes. And you can see one of those boxes, or one of those columns of boxes being pulled out of the metal container right there. Inside each box are 100 tiny tubes that contain tissue that can be used in genetic research. As a geneticist, I use these samples in my research, and I reach out to museums to get access to them. Once I have samples, including from all different types of animals from all over the world, I want to compare the coronavirus that we find in humans against the coronaviruses we find in other animals. The more similar two viruses are, the more likely that they're closely related. And we can use this information to figure out where a disease came from. Like before, this white line on top here is the DNA of the human coronavirus. When we compare the human virus to, for example, the coronavirus found in horses, we can see there are a bunch of differences. Each of these red dots indicate a different mutation or a difference between the human virus and the horse virus. Although this is slightly simplified, it's remarkably similar to what I look at when I compare genetic sequences. In viruses, mutations can change the shape of the virus's binding site. The binding site is the part of the virus that grabs onto other cells and allows the virus to infect them. If the binding site is a different shape, see how it's round here in horses, but a little more square for humans, that virus may not be able to grab onto a human cell. So even if that horse coughs on us, it can't hurt us. If we look at more coronaviruses from a bunch of different animals, we can see that the human version on top is most similar to the coronaviruses found in bats. The horseshoe bat RNA right here is most similar to humans. There's only one dot or one mutation that is different. In reality, the bat virus is about 96% identical to the human virus, and that's pretty close. Other viruses are only 50 to 75% as similar. This suggests that the virus that we find in humans today came from bats. So why bats, right? The first reason is bats are mammals. Humans are also mammals, so we get most of our diseases from other mammals like horses, cows, or even bats. Diseases that jump from animals into humans are called zoonotic diseases. And we can get them from being bit by an animal, being near an animal, or even eating animals. But if hundreds of other animals have coronaviruses, why didn't we get it from them? What makes bats so special? And bats are immune to most diseases, because they have a super strong immune system. Bats have actually evolved these extra genes or new mutations that allow them to make more virus-fighting proteins than other mammals. So maybe mutations can give us superpowers. It seems to have worked for the bats. When other animals get coronaviruses, they get sick and possibly die, which prevents them from giving it to us. But when bats get infected, they don't get a fever or a runny nose. They don't show any symptoms at all, which allows them to survive and possibly pass the virus on. Of course, bats didn't mean to give us coronavirus. In fact, we were invading their homes and hunting them and eating them. But knowing where coronavirus came from allows us to monitor the disease. We want to know, like, how many bats have this? Have they always had it? Do they show any symptoms? And we can also look at other diseases that the bats carry and learn if those can hop to humans. We also want to know about the places the bats live so we can understand how to keep both bats and people safe. Saving animals from extinction, saving humans from global pandemics, it's all pretty dark stuff. But genetics can also be used in cool or perhaps hot new ways. For example, we know the Earth is warming, and this means that species were adapted to cold weather, like polar bears, weasels, foxes, snowshoe hares, must be able to survive and adapt to warmer temperatures, or they may go extinct. What if we could change their DNA to help them survive? Maybe we could turn off the cold weather genes and turn on the warm weather genes to help these animals live normally in a warmer world. And this is what I'm working on now. But instead of studying polar bears, I'm looking at desert animals. Imagine you're in the middle of the desert. There is nothing but cacti for miles. It's 120 degrees out. That's real hot. And you're probably sweating, like a lot. And you're probably thirsty, but there is no water. As humans, we wouldn't last 3 days. In fact, I probably wouldn't even last a day. But cactus mice have already figured out how to live and thrive in hot deserts. These mice may never drink water or pee during their entire lives. And I want to know how they do it, right? If I can figure out what genes these mice are turning on or turning off to survive without water in the middle of the desert, then maybe I can edit those genes in animals that don't normally live in deserts so that they too can live in hot, dry areas, which maybe could save them from extinction. And we have a question.
Yeah, so how do we turn genes on and turn genes off?
Yeah, so I'm going to get into that. We're gonna talk about exactly how we can change genes. That's a great question. So first, we need to identify the genes that are involved in some of these cool things, like living in the middle of the desert without water. And we can do this in 3 different ways. I mentioned before that DNA changes or mutates slowly over time. If there are spots in the DNA that have not changed, though, we know that those spots could be really important and that those spots need to stay exactly how they are in order for the organism to survive. Scientists call this natural selection, and we can see it in their DNA. We can also compare the DNA of cactus mice or desert animals to the DNA of animals that don't live in deserts. The pieces that are consistently different may be the difference that allows these animals to live without water. Here we can see that the desert mice have these 3 different mutations that are present in all of the individuals, but not in any of the non-desert animals. And the last thing we can do is experiments. These are some pictures from my lab or our lab at the University of New Hampshire that simulate the hot and dry conditions of deserts. So we can basically turn up the heat or turn down the humidity. On the bottom are the little mouse houses, and on the top are a bunch of computers, each hooked up to a different mouse house. The computers measure how much water each animal is using and how hard they're working, just in their normal lives. These mice, they get all the food and water they want, they're chilling, they're happy, they even get little houses and they build nests. So we do this, we measure how hard they're working by measuring mouse breath. Yep. And we can test how the animals do when it's hot or when it's cold, during the day or during the night, and see what their genes are doing. And knowing the genes that allow these animals to live without water can also help humans. The human body is nearly 70% water. We can't live very long without water. In fact, we can only go about 1 day before we get really sick. More than 2 million humans die every year from dehydration or a lack of water, and many of them are children in developing countries. Genetics can potentially help us solve this global problem. Scientists can now edit genes using a technology called CRISPR. Yes, it always looks like it's spelled wrong, but it's actually an acronym. It stands for Clustered Regularly Interspersed Short Palindromic Repeats. And do not make me say that 5 times fast. That's a mouthful. And what this does is it cuts somewhere in the DNA that we want, and we can replace one gene with a different gene. So in theory, I could take out a cold weather gene or add in a warm weather gene to help animals survive climate change. So here's how it works. How does CRISPR know where to cut? We actually send in a guide with CRISPR, literally a guide to help CRISPR know where to cut. The guide RNA, which I've shown right here, matches to the place in the DNA that we want to cut. And once it matches, CRISPR, which is represented by this big blue circle, can cut out the target sequence of DNA, the spot we want to change. In addition to providing a guide, we also send in our edits. We send in the changes we want to make. This is shown as a separate piece of DNA. And really, it just kind of hangs out there while CRISPR is doing the cutting. Once CRISPR has cut out the piece of DNA we want to change, then that sequence is removed and the edited piece of DNA that we put in there, just floating nearby, can be plugged in. And voila, you've edited a gene. Easy, right? Gene editing is super cutting edge. Ha ha ha. But we're just starting to explore gene editing in mammals. CRISPR has already been used to reduce mosquito-transmitted diseases like malaria, and it's also being used as an option to prevent coral bleaching. Opposite of the mice that I study, corals don't do well in warm water. When the ocean gets too warm, corals kick out the algae that normally live with them. This causes the corals to turn white, like they've been bleached. Bleached corals are not dead yet, but they're under a lot more stress and they're at an increased risk of death. While some scientists are engineering new heat-resistant coral species or corals that can just live in warm water, others are working to find those genetic light switches that control how corals respond to heat. Once they find these light switches, they'll be able to recognize wild corals that are already adapted to heat. And then we can try to focus on saving the corals that are most at risk. Of course, this technology has also been proposed as a way to bring back extinct species. Think like dinosaurs and prehistoric mammals that used to live in North America, but have since gone extinct. Kind of like Jurassic Park, right? But it's not really that simple. Humans are known to be terrible at designing things. For example, nature makes wolves, but humans make whatever that little guy is. And trust me, I don't think he would last very long in the wild. So who's to say that our edits or our changes won't actually mess things up? Has anyone seen the ending of Jurassic Park? Let's just say it doesn't go well. And these are really important things for us to be thinking about, because the question now isn't, can we make changes to DNA of animals or of ourselves, but rather, should we? And this will ultimately be up to all of you. So that is the majority of what I've got for you guys today, and I would love to hear more questions.
Yeah, so we had one coming in that was backtracking a little bit to the mouse that doesn't pee.
Yes.
How do they excrete the nitrogenous byproducts of peptide metabolism?
That is a very detailed question. I would assume through other wastes. So they do, like they can secrete salts through their foot pads, and then they do poop.
Oh, okay.
They do poop. So they've got little baby poops.
There we go. And I was wondering, can we like produce proteins? It wouldn't be really artificially, I guess. It would kind of be natural. But like if we were using the bat, for instance, that had a really strong immune system, could we like extract the proteins from the bat? And would those, if like we put it in another animal, could it turn on other genes? If
Possibly.
they have genes?
Yep. So a lot of times what we're doing in genetics is learning from what's already out there. Like I'm looking at the desert species because they already do things we want to do. And the bats already are immune to all of these things. So there are a lot of people working on like, how can bats do this? We want to do this. But a lot of genes talk to each other. So, I mean, if your genome's 3 billion letters long, there's a lot of words, there's a lot of genes. And sometimes one gene depends on or uses another gene. So a lot of times these single genes need a network in order to function correctly. And that can be harder because the network that exists in bats might not work in humans. And so finding that balance so that it works without making humans into bats is really important.
Without like actually trying it or performing the CRISPR method on a certain genome, is there any way to predict what other genes would be affected or turned on or off if we did insert new DNA?
Yep. So scientists use computers to estimate or try to figure out the relationships between different genes. And we don't just randomly start changing genes. Usually you do those types of mathematical simulations in a computer first. And then we do it, most of my gene editing has been in fruit flies first. So you do small stuff first to make sure that it doesn't mess up all the genes, but hopefully only the one.
Right. And we had one question from Katrina asking, how rare is viral host switching? Is it the expectation or the rule, or sorry, exception or the rule?
I would say it's more of an exception, but as we are looking at evolution, the long history, how long viruses have been around, which is a long time, and how long hosts have been around, also a long time, we're kind of realizing that there's a balance between host switching, where a virus that goes into bats can then go into humans. That's host switching. Bats are hosts, humans are hosts, and instead of infecting both separately, it jumps between them. And that happens. But also, some viruses co-evolve with their hosts. So if a virus doesn't jump from bats into humans, but it specializes on just bats, it has different mutations or different binding site that is specific for bats, and then a different virus can evolve for humans. So there's a balance.
And we have a question from Marielle asking, could we use the antibodies from bats with coronavirus to help make vaccines for humans?
This is a good question, and I'm sure it's currently being explored. We do often use antibodies to figure out the pieces of DNA that we need to put into vaccines. But a lot of vaccines are also just pieces of the virus that can't actually hurt us. So we can definitely use that to help us save humans and protect humans.
All right, and then we had Molly asking, what is the role of CRISPR for traits that are many alleles of small effect?
Right, so that's super cutting edge and really, really hard. So like Molly was saying, and I mentioned earlier, a lot of these genes don't work as just one gene that changes your eye color. A lot of times it's 10 genes that affect your eye color as well as your fingernails or your height or I don't know, what time you wake up in the morning, all of these things. And changing one of those can impact all of those different traits. So that's super tricky. I haven't done this yet, but it is being done. And again, it takes a lot of simulation beforehand to figure out the relationships of one gene to other genes. Once you know the impacts of one gene on other genes, you can start to play with it. And one of the cool things that we can do in science is you can make a gene and you can model it, the protein that it makes in 3D. And then you can make your change and see how that changes what the gene looks like and what it does functionally for people or for other animals. And so you can see if you would expect your changes to impact multiple changes. But that's a really hard problem.
And do you have is it in any way, are a lot of genomes trickier than others when you compare them? Or is there kind of like a basis that you use or a reference DNA?
This is another great question. All genomes are pretty different. I'm really glad I don't study frogs. Frogs are really hard. They have really big genomes. And they're full of repetitive elements. So like lots of GA over and over and over again. And when things repeat a lot, it's hard to figure out what piece of your puzzle goes where because all the pieces of the puzzle look the same, which is really hard. Mammals do have what's called a conserved genome. So their DNA is mostly similar. And we have a framework of genes or a subset of genes that all mammals have. And you can use some of those genes that are the same in humans and in dogs or horses to compare directly between humans and dogs and horses. But humans also have genes that do completely different functions in us than they do in bats.
We have one question from Adam that says, do you think using CRISPR and other genetic alteration techniques to increase viral or bacterial resistance could lead to increasingly pathogenic and dangerous diseases?
Oh, absolutely. Absolutely. And this is why I think it's really important for more people to learn about CRISPR and to know what it is and what it can do. But they also need to understand the importance of diversity and natural selection and evolution, because if we start changing DNA, it's a slippery slope. Just like that little pug I showed you, it doesn't always go well. And the same thing is true for anything like diseases. We want to be really careful and thoughtful with how we are using these technologies. And right now, we don't really have the answers. There is a moratorium on using CRISPR on humans, but not all countries have signed on to this. So hopefully we can work through some of the ethical issues with this because it's pretty powerful stuff and we need to think pretty carefully before we move forward with it.
And so CRISPR is a relatively new technology, like you were just mentioning. So we might not know this yet, or maybe, I don't know if you've read a paper where this happens, but there is a lot of DNA that is what they call like the nonsense DNA that's or we just don't know what it does yet, or if it has an influence. Is there any evidence of like, if you insert a gene using CRISPR, insert some sort of change with CRISPR, that it would affect those like non-expressive areas?
Right, that's a good question. So You're right. Most of the genome for in mammals, for example, most of the genome is junk DNA. So it's non-coding, which means it doesn't make a protein. And so initially that seems like, okay, it doesn't really do anything, right? Some of those non-coding areas actually control the coding regions, how much we make of those of different proteins, or when they turn on and when they turn off. So not all of it is junk. There's also a cool thing, you could absolutely turn on or off things in junk DNA, but you'd have to find an area that could make a protein, but it would absolutely be possible using CRISPR. Yeah, basically sometimes mutations will happen and they randomly will hit an area of the junk DNA and you now have a new gene. Or a mutation could turn off an existing gene, and that could cause that gene to now evolve as junk DNA. It's no longer used, and that happens all the time.
And then Molly was wondering, what do you think about efforts to revive the woolly mammoth?
Right. So there's some really cool work going on. George Church's lab at Harvard is trying to revive the woolly mammoth by CRISPRing, cutting and replacing all the genes we know of that are in the woolly mammoth based on fossils and ancient, really old preserved hair samples. They've taken all the genes they were able to get out of that, and they're CRISPRing it into an elephant embryo to try to bring back the woolly mammoth. I think it's really cool. I'm hoping we don't have woolly mammoths like roaming in Montana anytime soon, but I still think it's really cool that we might be able to do this. I know one of the interests was looking at both the longevity, because elephants live a long time relative to other mammals, as well as what was it? There's an immune thing in elephants that's really neat, and as well as obviously how they produce hair, because woolly mammoths were hairy and elephants weren't. So there's a lot of neat things we can learn from woolly mammoths, but maybe not in Montana, maybe in zoos.
All right, let's see here. Do we have any more coming in? Will you just briefly, I want to get to this before we wrap it up at all. Will you briefly talk about kind of early influences in the genetics field for you, or useful skills or experiences that you've had that kind of pushed you into this field?
Yeah, so we were talking about that a little bit earlier. I had no idea what I wanted to do when I went to college. I switched majors like 6 times, and I finally found biology, which was a great fit for me because I loved animals. And I honestly didn't know what to do with that. I was originally a pre-vet major. I thought I wanted to be a veterinarian, and then I was working at a vet clinic and didn't love it. So I decided I was going to go to grad school in biology, and I was really into like behavior and studying wild animals. And I said, you know, the one thing I don't want to do is genetics. And here I am today, a geneticist, because my advisor, Joe Cook, at the Museum of Southwestern Biology, was an absolute inspiration to me. And I think he knew me better than I knew myself. And he realized that I would really like genetics because it explains so much, and it's so perfect and so simple, and you can see when an animal has this mutation, they do this, or which is so cool. And I felt a lot of the time when I was looking at behavior, I was like, I don't know why that animal's doing that. It's cool, but I don't know why. And now I feel like with genetics, there's a lot, you can explain so much of the world around you. It's not everything. There's still random behaviors and there's still randomness in the world, but genetics was a really neat, simple way to explain a lot of the cool things we see in nature. So I've had amazing mentors, and working with the Museum of Southwestern Biology at the University of New Mexico was the best ever. So.
And with your new job, working as the Curator of Mammals at the Kansas Biological Institute, or Biodiversity Institute, sorry, do you think, or do you kind of know what to expect with your day-to-day work? Are you going to be in the lab a lot?
Academia is always crazy. You never know what you're going to expect. I'm excited to curate mammals. That's kind of a dream job for me. The fact that I will be able to have students and teach, I'll be teaching classes like mammalogy. We do have someone else who runs the lab, so I won't be pipetting or moving liquid back and forth, but we'll be generating a lot of data. I work on really big computers called supercomputers to analyze all of that data, so I'll be doing that. And we're supposed to be building an international collecting program. So in theory, we will be traveling all over the world. My collection has amazing samples from a lot of Central America as well as Southeast Asia. And we have ongoing collecting in the Philippines and the Solomon Islands. And I hope to get back up to the Arctic. So we have to document our changing world.
Yeah, that's incredibly exciting. And there is one more question before I let you go. When a scientist extracts DNA from a sample like a fossil, is there a way to CRISPR that DNA as well?
That's a great question. So possibly. For example, when we extract DNA from a fossil, a lot of times the DNA is degraded. It's older, so it gets kind of chopped up into little pieces. And because the animal's not alive anymore, we can't CRISPR anything in that animal. But we can use that DNA to CRISPR into other animals that are alive. For example, the woolly mammoth and the elephant. The woolly mammoth is no longer with us, but we do know from fossils some of its DNA. And we can take that DNA and CRISPR it into elephants, which are a living relative of the woolly mammoth, to try to get hairy elephants.
All right, Dr. Jossi, thank you so much for spending your afternoon with us. You are a fabulous presenter, and we are so excited for you to start your new adventure as the curator of mammals. That's very exciting.
Thank you for hosting me. This is fun.
Yay. Okay. So I hope everyone has a really good afternoon. And if you have any additional questions, go ahead and send them to myself or the museum, and we will chase Dr. Jossi down and get the answers for you. Thank you so much, everyone, and we will see you next week.